started looking at this yesterday in lab. The data for yesterday's lab, by the way, is up on Angel in the lab data folder. Okay, so we're going to first, before we jump into the actual division process, we're going to look at aspects of what's called the cell cycle. And historically, initially this was simply divided into interphase and mitosis. Space here. The cell was either dividing or it wasn't, which seemed a reasonable approach until people really started studying, especially interphase, and saw that the cell really isn't just sitting there waiting to divide and then it suddenly divides. Interphase is actually a much more dynamic phase, and lots of stuff is going on. So people sort of subdivided interphase, as we learned more about it, into three parts: G1, S, and G2. And as we saw yesterday in lab, we divide mitosis into several subparts as well: prophase, prometaphase, metaphase, anaphase, telophase, which we'll get to shortly. But I want to focus us now, or at least for the next few minutes, on interphase. Because like I said, it's not just that the cell is sitting there waiting to divide and then it magically divides and then it goes back to just sitting there. Interphase is much more active than that, and it is an active part of the cell cycle. The other thing to kind of think about and realize is that when we kind of create these categories, it's more to sort of study it and to sort of categorize what's going on. Each one of these flows very nicely into the next part, so it's not as abrupt as this might seem. So in the G1 stage of interphase, as you see here, one of the main things that happens during G1 is that the organelles begin to double in either number or size, depending on what organelle we're talking about. But they copy themselves during G1, so that's a pretty active process. Okay, so again, the cell, even though it kind of, if you look at it under the microscope, it just kind of looks like it's sitting there, but it's actually doing quite a bit. So this is when all of the organelles, pretty much other than the nucleus, copy themselves. Okay, it's during this G1 phase. So if we're sort of doubling everything, then the other major thing that needs to happen then, if we're going to double everything in size, or double everything in number or size inside the cell, and the cell's going to have to get significantly bigger. So the other big sort of characteristic of G1 is that this is a significant growth stage for the cell. Essentially to accommodate that. So most of interphase, but especially G1, is very, very dependent on, making sure there's enough food available and things like that, so that these things can happen, so that the cell can grow, so that it can copy its organelles. And we'll talk about how the cell sort of monitors things and make sure everything's going sort of smoothly. The next big phase we've actually already studied, the S phase, is the DNA replication stage. So we've looked at that quite a while ago at this point, but we can now sort of tack this onto that previous step. Okay, so we've already looked at DNA replication, so we're not going to go through that again here, but this is, to put it where it kind of belongs in the cell cycle, it belongs to the S phase. So inside the nucleus, if we're doing a eukaryotic cell, all of the DNA will replicate using all of that process that we studied a month or so ago. And then once all the DNA is copied, it goes through a second G phase or stage, G2, where it will grow a little bit more, certainly not nearly as much as during the G1, but it'll grow a little bit more. And what they're talking about here, synthesis of proteins, it's basically proteins that are going to be needed to divide the cell. Those are the specific ones they sort of mean here. So a little bit more growth. And sort of a prep to divide. It's not a bad way to think about the G2 phase. And then, if everything's going well, it enters the M phase, the actual division phase, okay, which for us today and Monday will be mitosis. And then next Wednesday we'll look at meiosis. But luckily they both start with M, so we can just call this the M phase. And like I said, as we saw yesterday in lab, we are going to subdivide mitosis into those stages, if you will. Phase, and we will throw in prometaphase, even though we didn't deal with that yesterday. So they all make up mitosis, and very technically, mitosis is division of the nucleus. Very, very technically. However, we almost always include the division of the rest of the cell, but that technically has a different term. I'm running out of word space down here, so I'll put it here. And that is cytokinesis. They almost always go together. So the nucleus divides, and then the rest of the cell divides. Most of the time, but they are separable processes, especially in the laboratory. We can manipulate the cell to just divide its nucleus and not divide the cell, and sometimes vice versa. So these are separable events, but most of the time under normal circumstances, cytokinesis immediately follows mitosis, so we do kind of lump them all together most of the time. Although there are some really good examples where you just get mitosis and not cytokinesis. We're going to kind of ignore those exceptions here since we're kind of short on time at the end of the semester. Okay, so we want to have a good feel for what's going on in G1S and G2, and we'll spend a good amount of time looking at the mitotic aspect eventually as well. And then the rest of the table is just giving a couple of examples. Kind of like what you're going to calculate for that lab yesterday. How much time does the typical cell spend in these various stages? So Vicia falba is a bean, so it's sort of a plant example, and then Homo sapiens, fibroblasts are cells. A lot of them are in your epithelial types of tissue. They create extracellular matrix and things like that. So you can see that different cell types will spend different amounts of time in these various stages. This is not for memorization, it's just for illustration. And just some other examples of how long it takes a cell to go through the entire cell cycle. So I think I mentioned earlier, your intestinal epithelial cells divide about twice a day. Your liver cells once a year, or so on average. So it can vary quite a bit, from less than about half an hour up to a year. So different cells are getting different signals. We can link this back to this cell communication stuff that we were talking about. Right? So your liver cells only get that signal to divide once a year, whereas your intestinal and your stomach epithelial cells are getting that signal twice a day. Okay, so you can link this back to those signals, things that we talked about earlier in the week. Okay, so this is kind of just another picture of this. So again, rapid growth, not only the centriole, but other organelles double there. DNA replication, growth and prep, and then we'll look at these sub-category or sub-stages of mitosis as well. But before we get into the actual division part, we want to kind of talk a little bit about the rest of this cycle, because it's incredibly important that this process goes very smoothly and goes in this particular order around this circle or cycle. And it turns out that there are going to be important control points. They put a couple on here as well, so that the transition between, say, G1 to S is very tightly controlled, as is the transition from S to G2, and the transition from G2 to M, and the transition out of M back into G1. At each of these sort of transition points, there is what's called a checkpoint. And that's what I want to talk about for a little bit, is, again, how this is controlled, because your body wants to control this very, very carefully. Because what do we typically call out-of-control cell division? Cancer. Okay, so cancer is this process out of control. Or another way to think about it, the cell has lost control of its cell cycle. Okay, so if your liver cells are only supposed to divide once a year, and now they're dividing every 20 minutes, that's going to be a bad situation. Okay, that's one of the hallmark characteristics of cancer. Okay, so one way to think, a sort of simple way to think about cancer is that it is composed of cells that have lost control of this cell cycle. So I'm going to spend a little bit of time looking at how the cell controls this process. So one approach to this is to look at kind of what the cell is sort of dealing with. Actually, before we do that, that reminded me of one other thing. A cell that is not actively dividing is not technically in the cell cycle. So if we those liver cells, for example, that are sitting around not dividing most of the year are technically not in the cell cycle. Because if they're in the cell cycle, then they're doing these things. They're growing in size, they're doubling their DNA, or they're doubling their organelles, they're copying their DNA, etc. There is another part to this, and that's what that reminded me of here, called the G0 stage. That is the sort of true non-dividing stage. So liver cells, we want to keep that example, spend most of the year in the G0 stage, where they're actually not doing any of this stuff. Because if you're in the cell cycle, you're doing stuff. Okay? So if you're truly not dividing, like those liver cells most of the year, they're actually in this thing called G1, which is outside the cell cycle. Then when they get sort of tapped on the shoulder, hey, it's that one time of the year we divide, then they go out of G0 into G1, go through that cycle once, divide, go back out into G0. So some cells can go sort of out and in, depending on the signals. Some probably never go into G0, so your stomach epithelial cells are probably never in G0, because they're always being told to divide. So that's the one extreme. The other extreme is your nerve cells never exit G0, or at least we don't know how to get them to exit G0 very well yet, although people are working on that for obvious reasons. So once your nerve cells are established, they don't divide anymore, which is why damaging them is so damaging, because we can't really repair them very well. Although, like I said, people are certainly working on coaxing nerve cells back out of G0 into G1. So there is this whole sort of range of possibilities, but the big point is that there is this G0 idea when the cell truly is not in the cell cycle, it's not actively preparing to divide. Does that idea make sense? Okay, because most of the time people just think they can sort of hang out in G1 all the time, but that's not true, because things happen in G1. So if the cell's really not dividing, then it's not in G1, it's in G0. Okay. Question? Okay. So back to how this is controlled. Well, the cell, one way to look at it is that the cell is sort of looking and monitoring specific things at specific times. So during G1, when it's actively growing and actively copying all of its organelles, it's going to need things like nutrients, right? You need energy to do all that kind of stuff. So the cell's monitoring, hey, is there enough food out there for me to basically double my size and to copy all these organelles? Am I getting the signal to do that? We looked at growth factors, right? Is it getting tapped on the shoulder? Hey, time to divide. Okay, is it getting the right signal? As it's progressing through G1, it monitors that size. It doesn't want to get 3 times as big. It just wants to basically double in size so that when it divides, the cells are the same size. So there are ways that the cell has to sort of monitor how big it is. And the cell is always constantly monitoring its DNA for damage. Okay? And part of this idea is that if one of these things sort of goes away, the nutrients start running out, or there is some kind of DNA damage, the cell has ways to slow down the cell cycle and try and sort of either wait it out. So, okay, the nutrients are running out, maybe I'll just slow it down and we'll see if more food comes my way, kind of idea. Or, hey, there's a lot of damage, let me try and fix it first before we start copying the DNA, right? You probably don't want to copy damaged DNA, right? So let's try and fix the DNA before we start copying it. So these things that the cell is sort of monitoring are important because if some of these run out or there is, say, DNA damage or something, the cell can slow down the cell cycle to sort of accommodate. Okay, so it's not always, running at maximal speed. It depends on these kinds of factors. And the same thing up here for monitoring G2 into M. Again, it checks that the cell has gotten to the right size. Again, it's always looking for DNA damage, and it makes one more check that, make sure I copied everything. Because another aspect to this cycle is that it only moves in one direction. Okay? You cannot go backwards through the cell cycle. So if we think about these transitions as sort of gates or doors or tolls or whatever image works best for you, once you go through it, whatever the door, whatever closes behind you and locks. Okay, so if that door is the transition from G1 to S, and I'm in the cell and I say, all right, I think I'm the right size, I'm still getting told to divide, there's still enough food around, DNA seems okay, and I walk through that door, the door closes behind me, locks, and I don't have the key. Okay? So the cell ideally doesn't want to make a mistake, because if it's not big enough, and it goes into S, it can't say, oh wait, I made a mistake, I'm really not big enough to do this yet. Too late, you're done. Okay, you've made the commitment. Every time you pass through the door, you're committed to that next stage. There's no going back. And it's sort of like your washing machine dial, right? You can't spin it the other way. It only spins in one direction. So does the cell cycle. So that's another reason that these things are so important to monitor, because really, if you make a mistake, you're kind of screwed because you can't go back. Same thing with the S phase. If the cell makes a mistake and it didn't copy all of its chromosomes and it goes into G2, you can't go back and say, oh man, I forgot to copy chromosome 7. I'll go back and copy it now. Can't do that. Okay, so it is incredibly important that the cell really monitors this very, very carefully, especially the transition points. Because again, once you make that commitment, you've made the commitment. You can't go back. So it's looking for different things at different parts of this process, but it's under ideal situations, it's monitoring a lot of stuff, especially before it makes the commitment to go to the next stage. So then people, of course, wanted to know what's really going on here on a molecular level. What's really controlling the transition from G1 to S, from S to G2, etc. So people started kind of thinking, well, it's probably a protein or something, since proteins do most of the work in the cell. So you did some really kind of clever experiments to try and figure out what's going on. So This first set of experiments here shows an experiment where they fuse two cells together that are at different stages of the cell cycle. Okay, so this cell right here is already in the S phase, and they fuse it to a cell that's in G1. And then they sort of ask the question, what happens to the earlier nucleus? Okay? So when they fused it together, they found that the G1 nucleus gets pulled directly into S. It almost immediately starts to copy its DNA. Okay, so that gave them a little clue. All right, well, whenever it pushed this nucleus into the S phase, it does the same thing to that one. It draws it immediately into S. They did the same thing here. They did an M, a cell that was actively dividing. This was probably a difficult experiment to do, with a G1 cell. And same idea happened. The earlier cell, the earlier nucleus, was pulled to the later stage. So even though this hadn't gone through all of G1, no S, no N, or no G2, it got pulled immediately into the M phase. So whatever is controlling this, again, it's a absolute commitment. If this cell had already made the controllers for M phase, and that's going to work on this nucleus too, and it's going to skip everything in between. So these were kind of interesting experiments to show that there's got to be something in the cell that is pushing the cell through this cycle. So then, of course, they want to know what that thing is. And one part of the answer turned out to be proteins called cyclins. And they're called cyclins because, as you can see in this diagram, their concentration cycles as the cell goes through the cell cycle. It goes up and down. So this one is looking at a cyclin that's particularly important for the transition into M phase. And if we follow the blue line here, okay, this particular cyclin goes up, peaks at the M phase, and then goes back down to almost zero. Then as the cell goes through G1, S, G2, it's increasing, it hits the M phase at its peak, and then goes back down. So it cycles, hence the term cyclin. So that seemed to be pretty interesting, that we have this protein that they can monitor, that in this particular one peaks at the M phase, so this one probably is important for vision. Do I have a picture of the others? Okay. I don't have a picture of the other ones. But they also found others that peaked at other times. So there is one that sort of peaks at G1. There's another one that peaks during S. There's another one that peaks during G2. So this seemed to be kind of a nice story. That there are different cyclins and at least one for each of these sort of phases. So that seemed to be kind of a nice correlation, a nice possible story. But of course it begs other questions. So how does this work, and what are these cyclins really doing? So they eventually pieced together that the cyclins actually don't work by themselves. They work with another type of protein called a cyclin-dependent kinase. So we have cyclins, and we have cyclin-dependent kinases. Usually abbreviated CDKs. So we have a sort of a two-piece process here. So we have these kinases. What do kinases do? Phosphorylate things, right? We've been seeing that now for a couple of weeks. Kinasing things, phosphorylating things is an important way to control stuff, right? So we have these kinases that are going to phosphorylate things, but they are dependent on these cyclins. Because people showed that while the concentration of these cyclins varies, depending on where you are in the cell cycle, these CDKs, these cyclin-dependent kinases, are there all the time. They're in a decent concentration all the time. But that didn't make a lot of sense, because if they're there all the time, why aren't they working? But then putting it all together is they only work when their correct cyclin is at high enough concentration. So the two kind of work together. And don't worry, this picture is not actually quite as complicated as it looks. So we have specific cyclins working with specific cyclin-dependent kinases. And when the two when the right cyclin and the right CDK get together, the CDK works, it goes off and phosphorylates all kinds of stuff that seems to control this kind of march through this cycle. So it's kind of a clever way to do it and kind of a neat story. So we're going to look at two examples. We're going to look at the G1 into S transition and the G2 into M transition, but they all sort of work relatively in a similar way. Okay, so we saw, well, this is the second one that we'll look at, but there is another G1 cyclin, it's right here, whose concentration increases during G1 and sort of peaks towards the end of G1, and that's going to push the cell through that transition into S. So as that G1 cyclin concentration increases during G1, it hits a concentration at which it can attach to the G1 CDK. Two proteins getting together based on concentration. We have a value for that, don't we? What is that value called?
Km.
Km, right? Anytime two things get together, based on concentration, there's a Km value. So there's some Km value for that cyclin hooking up with that CDK. And when that G1 cyclin's concentration gets to that Km value, hey, now they can get together. When they get together, the CDK is now active. Active kinases phosphorylate things. And the targets of this G1 cyclin CDK complex include things like proteins that get the S phase started. So think back to getting DNA replication started, right? We had that sort of whole host of proteins that we needed to bind to the origin of replication and pull it open and all that kind of stuff. Those are some of the proteins that this active CDK phosphorylates, turns on, and the cell starts copying its DNA. So it's kind of a nice story, and it links back to things that we've studied earlier in the semester. So that's kind of the on switch, the rising concentration of a specific cyclin. Once it hits its Km, it hooks up with its CDK, activates the CDK, it starts phosphorylating target proteins that push it through that transition. It's kind of a nice story, I think. The other really cool thing about this is, so that's the on switch, but once we make the transition, then we need to turn that one off. The cool thing about it is the off switch is sort of built into this. One of the targets of this G1 cyclin CDK complex is a protease that breaks down that cyclin. Isn't that clever? So once it goes through that transition, the protease gets turned on, it breaks down the cyclin. If that CDK doesn't have its cyclin attached anymore, what happens to that CDK? It turns off. So it's a really kind of clever way to set up these kinds of transitions. So once you go through the transition, the cyclin is destroyed, turns off that particular CDK. Even though it's still floating around the cell, without its cyclin attached, it doesn't work. Okay, so this can be around all the time, but it's only active when its cyclin is in high enough concentration. Does that idea kind of make sense? So they all basically work in a similar way. So the G2 to into M kind of works the same way. It's a different cyclin and a different CDK, because we're at a different point along the cell cycle, but it basically works the same way. The M cyclin, as we saw here, oops, as we saw here, its concentration peaks right at that G2 to M transition, and it works basically the same way. Once that M-CDK, I'm sorry, there it is, MBM cyclin, sorry, gets to a high enough concentration, it hooks up with its mitotic CDK, as opposed to the G1 CDK. That starts phosphorylating things to get the process of mitosis started. So as we'll see a little bit later, it's going to activate things like, it's going to start phosphorylating histones, for example, to start the chromosomes to condense. And it's one of the early things that the mitotic cyclin CDK does, and that pushes the cell from G2 into M. And once again, how do we turn this off? You need to destroy the cyclin, and the same idea happens. One of the targets of the mitotic cyclin CDK complex is a protease that destroys that cyclin, so you get the off switch sort of built in to the process. So all of the transitions sort of work in a very similar way, through some cyclin and a cognate CDK, phosphorylating specific targets that push the cell through that transition point, from G1 to S, from S to G2, from G2 to M, etc. So it's kind of a neat system. And of course, we're just kind of glossing over, we're just doing this on this kind of surface level. There's a lot more to the story, but if you can get this basic aspect of the story, then we'll be in a good place. Does that kind of make sense, this idea? Cyclins and CDKs? Okay, so this is just a little more detail on that, the G1 cyclin CDK. So it turns out that there are proteins kind of already sitting on the origin of replication, but they don't work unless they're turned on. And there are sort of other proteins that control this. So this one here is called CDC6. This is a target for the first part of the story. So the cyclin CDK complex phosphorylates that CDC6. It falls off this protein. The protein's already there at the origin of replication and starts DNA replication, which is one example. Now, again, a couple of other things before we jump into the actual mitosis part. Is again, the cell is always monitoring important things, especially DNA damage. So I want to just real briefly talk about this, especially in terms of this protein right here called P53. If eventually you take my cancer class, say next fall, when I offer it, you're going to see that this particular protein, P53, is just extraordinarily important to the life of your cells. It is sort of the monitor of all sorts of stressors to your cells. For example, DNA damage in this example. I don't really have words to describe how important this protein is. It is the marker that's kind of constantly looking. Okay, is the DNA okay? Is the DNA okay? Are we damaging it? Is everything all right? It's constantly monitoring stuff like that. So that if there is a problem, it triggers, well, one of several possible outcomes. It can trigger, hey, it's not that bad, let's try and fix it. So let's slow down the cell cycle for a bit. Let's try to fix it. And it works through other proteins like P21 and things like that. We don't have to worry about the details here, but P53 is really the critical protein here. Because if the damage is too big, there's just too much damage. You were Bruce Banner and you got gamma irradiated and you turned into the Hulk. Right? Ideally, your P53 wouldn't let that happen. Okay? It would see all that DNA damage, and it can trigger, we talked about the other day, apoptosis, that cell suicide program. If there's too much DNA damage, that's what happens, that cell dies. Okay, so that's another possible outcome that P53 gets to decide about. So do we just slow down and try to fix it, or no, this is too much, we can't fix this, that cell needs to die. So that's P53. It's part of the job of this p53 protein. And the reason I'm making a big deal about it is because in many, if not most, if not perhaps all cancers, the gene that codes for this protein is mutated. Because if you're a cancer, getting rid of that would be a really good idea, wouldn't it? Because then the cell doesn't care about DNA damage. It just keeps going through the cell cycle. And that is a very pro-cancer kind of thing. Because cancer starts basically through mutation. So if you can up the mutation rate, that's a very pro-cancer kind of thing. So cancer cells almost always get rid of that protein in one way or another. And that really opens up a lot more of the cancer phenotype because the cell no longer has the ability to monitor these kinds of problems. So it doesn't care about those things, so it's just rushing through the cell cycle with really no way to stop it anymore if you get rid of p53. So, like I said, if you take that cancer course with me next fall, we talk a lot about that particular protein in the context of cancer. And this is just another figure for the mitotic one. We already looked at that. We'll see shortly, maybe not today, maybe on Monday, that another one of the targets of this mitotic cyclin CDK complex is this thing here called the anaphase promoting complex, which, as its name suggests, is a protein complex that promotes anaphase, that pushes the cell through prophase, prometaphase, metaphase into anaphase. Okay, so that even that level is under the control of these cyclin CDK complexes. So we'll talk about more of that once we get to anaphase. Okay, so these are other things that are happening. So it targets proteins that break down the nuclear envelope, that cause chromosome condensation, that start the spindle forming, that targets proteins for degradation, which is part of what the anaphase promoting complex is going to do, as we'll see. So these cyclin CDKs are really the orchestrators for basically the entirety of the cell cycle, but in conjunction with things like p53 that's doing the monitoring of other aspects and can interact with these and say, no, we need to slow down for a little bit or what have you. So again, these are some things that especially p53 is looking at. So what's the environment like? Again, we talked about that a little bit already. Is there enough food? Is there the right signals? DNA damage is looking for constantly. Before it gets out of the S phase and goes into G2, it checks and double checks and triple checks that all of the DNA has been replicated. Because again, you don't want to get into G2 and say, oh shoot, I forgot chromosome 12. That would be bad. And then through G2, again, it's looking for DNA damage, and it's checking again that it copied all the DNA. Not that it could go back, but if it had forgotten to copy chromosome 12, that cell would probably die. It would be forced through apoptosis. And then throughout the M phase, it's not going to let anaphase happen until all of the chromosomes are hooked up to microtubules, and they're all exactly at the center of the cell. So there's lots of stuff that the cell is monitoring throughout the cell cycle to make sure that it's running smoothly and correctly. Okay, because again, the alternative is bad. The alternative is cancer. Okay, so that's a little bit about the cell cycle. This, again, is another topic where you could spend an entire semester just looking at the cell cycle and mitosis. We're going to do it in 2 class periods. So we are just looking at sort of the surface of this, but there's a lot going on. Okay, so that's a little bit about the G1S, G2 part of the cell cycle. Everyone feeling okay? Okay, so let's at least get into the beginnings of M phase. Okay, so the cell has made the decision, everything's okay for the transition from G2 into M. So one of the early things that the mitotic cyclin CDK is going to do is to start the chromosomes condensing, and that's part of what's being shown here. So as the cells had gone through S phase and copied their DNA, the sister chromatids, so I guess we should throw that in. Okay, so let's say this is number 15, it doesn't matter. When there's only one copy of it, call that a chromosome, and the cell goes through the S phase, the chromosomes get copied, right? And the two copies initially are held together. At this point, the two identical copies of chromosome 15 are called chromatids. And typically, the two identical ones are also sometimes referred to as sister chromatids, since they're identical. And these, as you can see here, are held together by a protein. The two copies, the two chromatids, are held together by a protein called cohesin. Okay, that kind of keeps the two chromatids together. Just to set us up for the future, that's the protein that the anaphase promoting complex is going to break down so that the two copies can be separated. But more on that later. Okay, so that's kind of set up during the S phase. Here we are in now the earliest part of M, so the cells made the transition from G2 to M. One of the first things that starts happening during prophase is that the chromosomes start to condense. Right? Remember the cells that we looked at yesterday? We distinguished interphase from prophase because we could start seeing the chromosomes in the nucleus of a prophase cell, right? We can do that because they start to condense. So think back to chromosome structure, right? We talked about all those levels and histones and nucleosomes and 30 nanometer filaments and all that kind of stuff. At each level, it gets more compact and therefore more visible. So that is triggered in a couple of ways. One is the addition of a protein called condensin that starts establishing those higher level orders of structure. And the other thing that's happening sort of simultaneously that gets this process started is that histone H1 is phosphorylated by this M-cyclin CDK complex. Phosphorylating H1 is another trigger that the chromatin, the DNA plus protein, starts to condense, eventually into a visible form where we can see the two chromatids fairly well under a light microscope. So this is what we kind of looked at yesterday. So in an interphase cell, we can't really see the chromatin very clearly. But then by prophase, through the work it partly started by phosphorylating histone H1 and the work of that condensin protein, chromosome starts to condense into a visible, much more compact, much more visible form. And it seems to do that simply because if it's smaller and denser, it's going to be easier to move around. My analogy is always think about when you're going on a trip. There are sort of two ways that you could bring your clothes with you, right? You could just kind of hold them all in your hands somehow and hope that you don't lose any. But most of us usually pack them in a suitcase, right? It's sort of the same idea. Now, why do we do that? Because then we just have one thing that we're dragging behind us and it's easy to move all that stuff. Well, the same thing here. Remember how long these DNA molecules are? If we kind of compact them and put them in a suitcase that is the chromosome, it's going to be easier to move them around than if we're trying to drag this, zillions of feet long piece of DNA. So that's my little analogy for that. So that seems to be why the chromosomes condense into these much more compact and therefore much more visible configurations. So we talked about chromosome structure before. I just want to remind you that that constriction point there is called the centromere. That's going to be important for us later because that's where the microtubules are going to attach eventually. So that's that indentation there. Where the little arrow is there, right here. That's the centromere right here. Okay, so now we're well into prophase, right? So inside the nucleus, Chromosomes are condensing because H1 is being phosphorylated, because condensin is working, among other things, but those are two important things. Meanwhile, still inside the nucleus, the nucleolus fades away because ribosome production shuts down as the cell divides. Remember, the nucleolus is the site of ribosome production. So that production shuts down, so there are no more ribosomes being put together there, so that sort of fades out, so you don't see that anymore during division. But prophase, the nuclear envelope is still there. Meanwhile, out in the cytoplasm, the centromere, the microtubule organizing center, so we need to hook this back to cytoskeleton and microtubules. Right? Microtubules need the centrosome as its organizing center. Back during G1, that copied itself, so now there are two centrosomes, so there are two organizing centers. And while the chromosomes are condensing inside the nucleus, the two copies of the centrosome out in the cytoplasm are moving apart. They're moving to opposite poles, is the usual term, or opposite ends of the cell. And between them are growing microtubules that we're going to call the spindle microtubules. There's our nucleus with our chromosomes condensing. Now we have our centrosomes moving to opposite poles, and remember we have two little centrioles inside. And what's growing out are sets of microtubules that collectively we're going to call spindle microtubules. Some of them are going to attach to the chromosomes, some of them are not, some of them will attach to each other. We'll see that as we move through. But for now, the spindle apparatus, the microtubules that are going to move stuff around, are being established out in the cytoplasm while the chromosomes are condensing inside the nucleus. Now, of course, we kind of ignore all the other organelles in the cell. They're there, of course. There are still mitochondria and ERs and Golgi and that kind of stuff, but we kind of ignore them and focus on the microtubules and the chromosomes during mitosis. But they are still there, and we'll deal with them at the end for cytokinesis. Okay, I think this is a perfect place to stop for today. Okay, so we've gotten through prophase. Okay, so on Monday we'll pick up with prometaphase, finish up the process of mitosis, and then